So welcome everyone to the joint prospective European drug-induced liver injury network and cost drug-induced liver injury seminar with EUROTOX. So we are going to cover today three speakers, one from the preclinical setting, one from clinical, and one from the FDA and regulatory. And so we'd like to run these three seminars looking at trying to run each of the discussions back to back and then have a round table at the end. For this seminar session, would it be possible to post any questions that you have in, so when you click onto the link, there's a EUROTOX homepage website. And on that, there's a chat function. And from there, we can screen and monitor the questions that people have. So it'd be great if people could put their questions into there. And so with that, I'd like to start with the first speaker, which is Dom Williams, who's a director at AstraZeneca. And he's the target organ strategy lead for preclinical hepatotoxicity assessments and leading a team of hepatic safety scientists. And so with that, I'd like to just hand over to Dom.
Hi, Tom. Thanks very much. I'll just share my screen. Okay, hopefully you can all see that. So firstly, I'd like to thank the organisers for inviting me to this really interesting session. I'd like to talk about new in vitro models of drug-induced liver injury, but I've changed the title slightly to talk about integration of these models into preclinical DILI risk assessment and how we're able to use the latest in vitro models to conduct risk assessment. Tom, just a sound check. Can you see that okay?
Yeah, everything's through.
Okay, brilliant. Thanks very much. And as Tom says, please write any questions you have. I'll try to address them throughout the talk, but if not, we'll get to them at the end. So as a structure of my talk, I'll very briefly introduce DILI. I'm sure it doesn't really need any introduction, but just talk a little bit about the pathogenesis and the mechanisms of drug-induced liver injury. But then do a bit of a dive into what we do within AstraZeneca, how we've set up an in vitro hepatic safety toolbox, and how we've developed this into using assays and PK information to conduct an overall Bayesian DILI risk assessment, and also how we've incorporated new models, hepatic spheroids, into this machine learning model. But obviously, the predictivity goes hand in hand with the actual assays that we're using. So how do we make those assays better, higher throughput, more sensitive, more specific? And also how do we address different DILI mechanisms? So I'll focus in on bioactivation-induced cytotoxicity, and then finish with a little bit of a case study about how we do risk prediction with novel in vitro models for drugs that we don't actually have a lot of data for. And I'll use this example with the covalent inhibitor drugs. So drug-induced liver injury, we know it's a leading cause of acute liver failure. It's got a high sickness and death rate. It's the main reason for late-stage termination or withdrawal of drugs. There's a number of drugs which cause hepatotoxicity across a number of DILI registries, and a significant number of drugs have been withdrawn or given a black box warning. It's certainly it's been seen recently in clinical trials, with a number of clinical trials being stopped in clinical development. And one of the more major recent cases is the stopping of the Janssen BACE inhibitor program due to drug-induced liver injury. And so broadly, the physiological function of the liver is clearance, and anything which interferes with clearance has the potential to cause hepatic stress. So we have drugs being taken up into the liver, they accumulate, and then they're undergoing metabolism. And these processes have the potential to interfere with various different normal liver functions. So, for example, the mitochondria, we could get inhibition of biliary efflux. We can get formation of reactive metabolites, which cause chemical stress. We can get activation of the immune system and a number of different mechanisms. And broadly, these can result in multiple varying phenotypes by which patients present with drug-induced liver injury with varying clinical and histopathological features. But really, the DILI patients provide these mechanistic clues that allow us to go back into the laboratory and look in vitro models to see maybe what we missed the first time around. And so the AstraZeneca hepatic target organ strategy, essentially there's three main parts to it. We start off with a simple risk assessment, which consists of assays that are used relatively high throughput, give reproducible data, which we're able to combine into a risk assessment model. But then we extend that by conducting bespoke understanding, which is very much on a case-by-case basis, depending on the drug project. And this is really where we start using primary human and animal hepatocytes and more complex models, such as multicellular spheroids or liver on a chip. And then we combine these two risk assessments into a tailored drug-induced liver injury prediction, by which we give advice to project teams. And I think the main aspect of this is that we want to do quantitative translation from in vitro, understanding mechanisms, incorporating these into the risk assessment across species to predict liver injury in humans. And so I'll take you through this process about how we do this in AstraZeneca. This is the AstraZeneca hepatic safety toolbox, and this is the simple risk assessment which I mentioned previously. And this consists of 7 different inputs, and these can be assays such as BSEP inhibition, the bile salt export pump inhibition, cytotoxicity in glucose and galactose, which assesses for mitochondrial toxicity. In red, I've incorporated any novel model here. So here we have a very slow proliferating clone of HepG2s into a spheroid model, which is a 4-day assay. The THP1 cell line is very good for generic cytotoxicity, but then we combine these assays with predicted Cmax, C-log-P, and a binary understanding of bioactivation. So, for example, can a drug undergo bioactivation, yes or no? If it's a yes, it adds on a certain level of risk in the Bayesian model, and I'll talk about that in a little while. After the simple risk assessment, we then enter lead optimization phase, which is really where earlier toxicology studies are performed, where we get an understanding of sort of whole animal responses. But at the same time as these studies are being undertaken, this is where we do bespoke risk assessment. So this is using primary hepatocytes, plus and minus cytochrome P450 inhibitors. We can look at use primary hepatic spheroids with non-parenchymal cells, Kupffer cells, and endothelial cells. We can look across preclinical species, primary hepatocytes, integrate DMPK safety endpoints, and also more complex models such as liver chip. And then after that, this progresses then to candidate drug investment decision, which is a large investment for kicking off the regulatory studies. And so we have this toolbox of routine and bespoke assays that influence chemical design and decision making. But these assays are monitored continually for performance versus emerging novel systems, because we want to make want to have the best sensitivity and specificity for any model that we incorporate into this toolbox. This is the probability data visualization in the Bayesian machine learning model. This has all been published, so please take a look in Chemical Research and Toxicology. Sorry, someone just went off mute there. So the data from each of these assays has been gathered for 96 compounds, 33 no DILI compounds, 40 medium DILI risk compounds, and 23 high risk compounds. Each of these endpoints is then weighted for its predictivity. And so things like Cmax and bioactivation tend to have a heavier weighting versus the other inputs, even though they all add to the predictivity. And then the IC50, the assay values, are multiplied by the weighting and then added together to give a continuous risk score. And I'll just play the video again. You can see this curve, the profile changing shape. Essentially, this variation captures the uncertainty, because you can have some assays which suggest the drug is completely safe and some assays which predict the drug may be toxic, and this gives a variation into the weighting on the variation on the curve. And here we have compounds which range from clinically safest. These are coloured in green. These are low DILI risk, going to medium DILI risk, coloured in amber, and high DILI risk, coloured in red. And these severity indexes they're binned into three categories, but they're based on the NIH EUROTOX severity index. And we see this right shift of probability of DILI becomes greater, and this aligns with the DILI severity categories of these molecules. And as you can see here, the statistical thresholds and proportion of data are calculated. And given this uncertainty in the prediction, the breadth of the profile, often they can span risk threshold. So really, we say that there's no real threshold. Obviously, no DILI versus minor DILI, there is a bit of a threshold there, but broadly, DILI is on a continuum, being differential severity in different individuals. And I'll just play that one more time so you can see. Here we have all the different data from the different assays and all the different compounds in the validation data set. And often paired DILI compounds give confidence as to whether an assay is good at predicting DILI. And these compounds have high Tanimoto chemical similarity. And so buspirone and nifazodone are very similar in chemical structure, but have very different clinical risk profiles. Similarly with ambrisentan, bosentan, and sitixantan, they get more clinically dangerous and hazardous as we go down the page, down the slide, and consequently the risk profile is seen as increasing. And so this is what we use in AstraZeneca at CDID for understanding part of the risk of a molecule. So here we have in blue a new AstraZeneca compound. Here we have the summary stats. This profile in white is where the average DILI probability for severe drugs is, and so we really don't want to have our profile overlapping with that. Here we have the assay data. Here we have the continuous risk or probability of risk of DILI, and we have the AstraZeneca compound in blue, and we can see which compounds it associates with in terms of its similarity of assay data. Probably the most interesting part of this is this statement of risk at the bottom, and this is really where we incorporate any bespoke understanding of risk, where further mechanistic data is generated, and the two things are combined: a very formalized quantitative risk assessment versus slightly lower throughput models, but using primary human hepatocytes. And I'll take you through some of these assays. So about 7 years ago, it started becoming obvious that hepatic spheroids were of real benefit because they retain their cytochrome P450 functionality much longer than hepatocytes in 2 dimensions. And they also demonstrate a gain of function. So transporters were functional for longer. There was a longer usage period, so you could understand whether sensitivity increased over time. You can also do co-culture, which provides additional mechanisms of toxicity versus hepatocytes alone. And some of the endpoints, ATP was a very sensitive endpoint in 3D. Interestingly also that you can get clinical pathological endpoints within the spheroids. So if you inhibited mitochondria, you could get steatosis occurring and phospholipidosis and what have you. And broadly, publications at around about that time confirmed that these functional capabilities were better than cells cultured in 2 dimensions. we published a joint paper with Genentech and Insphero. We did an analysis of the human hepatocyte spheroids with 69 DILI positives, 41 DILI negatives, found that there was greater sensitivity in 3D. And this was using primary human hepatocyte spheroids plus 10% non-parenchymal cells via the Insphero hanging drop method. And we use this across AstraZeneca for about between 2015, 2016 for about 216 compounds. And we also fed into the MIP-DILI IMI1 consortium, and really interesting findings there suggest that the 3D models reflect the original donor proteome very well. They're robust and sensitive, but they do show inter-individual variation. So further confidence building we got was we looked at cross-species spheroids in human, rat and dog for paracetamol toxicity. And the expected order of sensitivity from the literature is human is the most sensitive, dog is intermediate, and rat is quite resistant because it has a much lower cytochrome P452E1 activity. And broadly, we found the sensitivity of the spheroid models to be similar in that human were the most sensitive, rat was in the middle, dog was in the middle, and rat was the least sensitive. So it gave us confidence to continue using these spheroid models. What we've done internally is we found when we use spheroids, what we used to find with hepatocytes cultured in 2 dimensions was because the cryopreservation procedure made them somewhat delicate. When we treated with paracetamol, we used to get concurrent cytotoxicity and glutathione depletion, both occurring at the same time, suggesting that cell death was causing the glutathione depletion. But what we found when we use spheroids rather than 2-dimensional culture is we get the correct mechanistic order. So we can see in the complete absence of cytotoxicity, we get quite significant glutathione depletion. And this also occurs at different time points. We have a longer usage period as well for the spheroids. So again, gaining further confidence. And as I say, this was an internal model, so we worked with Insphero, but then we were able to gain understanding and take this model in-house. Some of the stuff we've done previously, just to give you an idea of how we use spheroids within AstraZeneca, is we found that across my group, the different scientists were actually producing spheroids that were different sizes and different capabilities. So we wanted to normalize this somewhat. So we used a Bravo machine in order to get automated production of spheroids across seeding, dosing, sampling, picking, pooling, all of that. And we found it gives us much decreased variation. And so, again, confidence that there's less user variability in there. But we can go to quite phenomenal numbers of spheroids now, depending on what type of experiment we want to conduct. Broadly, I'll talk about a few of these different areas. I'll talk a bit about bioactivation-driven toxicity. So we use cytochrome P450 inhibitors to look at whether metabolism affects the health of the cell. We're venturing into transcriptomic-based safety predictions, again using spheroids, but running transcriptomics and then looking at whether the omic profile can give an indication of DILI risk. We're also reverse translating biomarkers into spheroid models. So what we found is that modelers find it quite beneficial to build a model based on the same endpoint that's used in vivo, preclinically in preclinical species and in clinic. And so we're looking at using those same biomarkers in vitro models. So here we get an understanding of how ALT is released within that in vitro model. And also understanding a little bit more about the mechanism. So do we get glutathione depletion first, or do we get cytotoxicity? I'll talk a little bit more about that. And so here we have our bioactivation-induced cytotoxicity. So obviously P450s can bioactivate drugs to reactive metabolites, which may or may not be toxic to the hepatocyte. So this is a representative figure graph whereby a compound causes cytotoxicity in the absence of ABT. But then when we include this ABT, which is a non-specific cytochrome P450 inhibitor, if we see decreased toxicity, this indicates that a reactive metabolite is not being formed and that the cytochrome P450s were responsible for that reactive metabolite formation. In some cases, we actually see enhanced cytotoxicity, indicating that clearance is actually a mechanism of detoxification of the parent compound. We often see this with oncology agents. And so the parent compound is more toxic than any metabolites that are formed. We're able to go through our bank of DILI positive compounds and look for where we see this disparity. So for some compounds, we see a decreased toxicity in the presence of cytochrome P450 inhibition. And for some compounds, we see increased toxicity in the presence of cytochrome P450 inhibition. So it allows us to look at risk when we both have CYPs active versus inactive. So next, I'd like to talk a little bit about the risk prediction using the covalent inhibitor drugs. And so I mentioned we do a simple risk prediction in the first instance. And so what tends to happen when we look at covalent inhibitor compounds that have this is their mechanism of action, these tend to be predicted as high risk in our simple Bayesian DILI predictivity model, because we use an oncology-derived cell lines, because these are mainly oncology drugs anyway. This is their mechanism of efficacy, so they tend to kill the Hep G2 assays, but also bioactivation is set to default because the covalent inhibitors have an electrophilic warhead. Bioactivation is set to yes as a default. So here's an example using afatinib. This is an electrophilic alpha-beta unsaturated carbonyl compound. Bioactivation is set to yes within the Bayesian model, and we get an idea of relatively high, medium to high risk. But then when we use primary human hepatocyte spheroids cultured for 10 days, here we get a change in the risk assessment, because here we can see that the cytochrome P450 inhibition doesn't affect the cytotoxicity profile, but we also have quite a significant safety margin towards the Cmax. In this case, nearly a 40-fold difference between where the compound kills hepatocytes versus the maximum concentration that's going to be achieved in the human. So that can make us relax a little bit that we've actually got a decent safety margin. And so this is one way to integrate a simple risk assessment assay with an experimental assay using primary human hepatocyte spheroids. And so what is the concern about that we have with covalent drugs? Well, they obviously have an electrophilic warhead because they're designed to fit into a pocket and bind to, in this case, cysteine. But obviously we've got the other part of the molecule as well, which has the potential to undergo cytochrome P450 mediated bioactivation. So in this case, we have in what may happen in any one molecule, we may have two electrophilic centers, one being the electrophilic warhead and one being the electrophilic metabolite. So how do we pull out what's dangerous versus what's efficacious? Some of the knowns here is the reactive metabolite. We know it's an electrophile generated through metabolic bioactivation, but it has an unknown level of reactivity, and it may or may not be detectable by MET-ID, but it could cause organ damage or immune-mediated adverse events. The warhead is a weak electrophile. It's designed to covalently bind to a cysteine in the target protein, but we can use, because it binds to glutathione, we can use the glutathione reactivity assay to estimate the compound half-life in the presence of glutathione, but there could be off-target binding to other cysteines, which we need to know about. And so broadly, we split this into the areas that we need to be concerned about: warhead reactivity, metabolism-driven activation, but also take into account the patient centricity of this. We know the warheads bind to cysteine. So does this binding to cysteine actually cause glutathione depletion and then consequently cause cell death? And do we get increased sensitivity upon chronic exposure? The metabolism-driven bioactivation, we know reactive metabolites could be formed. if we get P450 inhibition, does that affect cytotoxicity because it keeps the warhead with the cells and the proteins for longer? But really, MET-ID helps us pull apart these different structural alerts. And then the patient-centric assessment, really, we need to understand whether the target protein varies across patients, what are the comorbidities and co-medications, but is it really for an unmet medical need where there are no other drugs there? So a higher risk assessment is okay. And so this is an example, a compound called zanubrutinib, where we've used primary human hepatocyte spheroids. Zanubrutinib is an oral inhibitor of Bruton's tyrosine kinase. It causes ALT elevation in below 5 times upper limit of normal in about 30% of patients, but it also causes quite severe ALT elevations in less than 1% of patients. But mechanistically, when we look at this in primary human hepatocytes, we can see on spheroids in day 1, we don't see much cytotoxicity, but we do see a certain amount of glutathione depletion occurring. But over 10 days exposure and 2 repeated dosings, we actually get this swapping over where the cells become slightly more sensitized, so die earlier, whereas the glutathione levels, although they vary somewhat, have a much more drastic depletion when a certain threshold is crossed. So it helps this understanding of the different risk profiles we may gain. What generally helps is an understanding of integrated MET-ID analysis. This is an AstraZeneca compound in this case. And here we saw that in the presence and absence of P450 inhibition, there wasn't really any change in cytotoxicity. But MET-ID really helped in this case because we were able to look at MET-ID both in the presence and absence of aminobenzotriazole cytochrome P450 inhibition. And you can see that we see both glucuronidation and glutathione addition to the warhead. This takes place even in the absence of P450 metabolism, as you would expect, because the warhead doesn't require metabolic activation to bind to glutathione. But all the oxidative metabolites simply weren't there when we inhibit cytochrome P450. So that gave us further confidence that inhibition of cytochrome P450 had no effect on cytotoxicity in this case. And broadly,
sorry, I'm just there's 5 minutes to go.
Brilliant, thanks, Tom. Nearly there. So this has allowed us to do risk assessment in relative absence of a great deal of information, particularly with novel in vitro models. So we've taken a number of different covalent inhibitors, each of which have an acrylamide warhead. And obviously, these have been used in patient populations with life-threatening diseases. And they're all obviously kinase inhibitors, some of which are FDA approved drugs, but some of which are competitors, which I've had to blank out a little bit. But this figure, the y-axis is the percentage of patients with an ALT less than 5 times upper limit of normal, and then the shape of the sort of point on the graph actually is where ALT is greater than 5 times upper limit of normal in patients has been seen. And we calculate a safety margin in spheroids, which is basically the EC50 at which spheroids see cytotoxicity divided by the Cmax. And so what we can do here, we've got these covalent inhibitors with known clinical experience, even though it's relatively recent, and we can position an AstraZeneca drug on the vertical line on the x-axis and really see where our AstraZeneca drug associates with these different covalent inhibitors to give us the first indication of where that risk may actually lie. And this can lead to us, we've put together quite detailed decision trees that allow a really robust framework to assist risk assessment. It is quite complex. I won't go through this, but I'm sure we'll be able to access the slides after the talk. You can see that we make decisions, risk assessment decisions on each stage of the decision tree. And these don't have to be set in stone, but obviously the more compounds we put through this, the better idea of the risk we are facing with each of the compounds. And so things like IC50 in the absence of ABT versus smaller than the IC50 in the presence of ABT, all lead to these different decisions as we go down. But the detailed decision tree goes into some quite complex mechanistic issues with covalent inhibitors. And so broadly, I'd just like to summarize. We have a place for both simple models and really complex models within the drug safety pipeline. 2D models, we want high throughput, little variation, but we want fast turnaround, low cost, and reproducibility. And then we move into simple 3D models that can be primary human hepatocytes, better metabolic function, however, not generally quite as higher throughput as the 2D models. But then we also have the complex 3D, multicellular 3D and microphysiological systems that allow us to have cutting edge physiological mimicry. And so broadly, to summarize, I've given you an overview of preclinical simple risk assessment versus bespoke risk assessment in DILI. Simple higher throughput models can be used for routine risk assessment and can be validated against hundreds of known DILI risk drugs, but lower throughput complex models have to be used for more bespoke relative risk assessment to 1 or 2 compounds. Spheroids certainly enable integrated toxicity and metabolism over time. But each simple and complex models have a position within drug safety assessment. But really what we're faced with at the moment is AstraZeneca's pipeline has greater than 50% of new modality medicines, such as dendrimers, lipid nanoparticles, antisense oligos, all sorts of things, which don't rely on cytochrome P450 capability anymore. So we need to make sure that we're sort of future-proof, that we have the appropriate in vitro models in place, which may mean multicellular uptake and exposure, and PK may affect the safety endpoints. We've had some of the drugs that are taken up by Kupffer cells and endothelial cells, which have a massive effect on the safety and the cytotoxicity towards hepatocytes. Also, in vitro models may just lack appropriate proteins. We found for drugs, PROTAC drugs, that cryopreserved human hepatocytes just don't contain the ubiquitin E3 ligase, so there's going to be large deficiencies when we come to do safety assessment of PROTACs. And so, yeah, hopefully I've given you a bit of an overview. The majority of this presentation was done by 2 members of my group, Sarah Ambertson and Barvik Chuan, so a real great thank you to those guys. But this is a collaboration between Inspiro, Genentech and the IMI1 consortium, MIP-DILI. So thanks very much. That's me done.
Thank you very much to Dominic Williams from AstraZeneca. And I'd like to encourage everyone to type any questions that they have to any of the speakers who will be with us today into the chat box that you will find at the official EUROTOX website, where you accessed most probably this meeting. So if you have any questions to any of the 3 guests that will be here today, please go ahead and type them there, or you will be able to ask in person after the presentations are done. And now, just to get all the information that we wanted to share with today, we invite James Deere, who's a professor of clinical pharmacology at the University of Edinburgh, and who's leading a research group exploring the role of microRNAs in toxicology and cell-to-cell signaling, to speak with you today too. So, Professor Deere, there you go.
Thank you very much. I'll just share my screen.
Perfect. We can see our slides, but they're in the presenter's view.
In the presenter view. One moment, sorry. 3rd time lucky.
Yes, indeed.
Fantastic. Thank you very much. No matter how many times I do Zoom talks, I still struggle with the technology. Well, thank you very much for inviting me to talk at this very interesting symposium. Now, I'm James Deere. I'm from Edinburgh. And today I'm going to talk about paracetamol and the use of new biomarkers. Now, one of the things I've always failed to do in almost every talk I've ever given is have slides that have paracetamol or acetaminophen, but not both on. So I apologise from the start that I've noticed I've called it acetaminophen on the first slide, but as you all know, paracetamol and acetaminophen are the same thing, just which side of the Atlantic you're on. So Dom in the last talk covered this very well about drug-induced liver injury, and I don't think I need to talk to this audience about why it's important and that it's the leading cause of acute liver failure and a major reason for drugs being withdrawn from the market. But one thing I would like to say is that the most common cause of DILI, at least in the Western world, is paracetamol overdose. So why did I choose to study paracetamol overdose in the first place? Well, there's two reasons I think paracetamol overdose is important to study. Firstly, it's a model of acute organ injury in humans. We understand the mechanism well. Our mouse models translate well into humans, and it's a model we can use to develop biomarkers and new treatments. And secondly, as we'll come on to, it's a common clinical problem, particularly here in Britain and actually particularly here in Scotland. So just to remind everybody about the mechanism of paracetamol toxicity, paracetamol in a therapeutic dose of 1 gram is safe. It's predominantly metabolized by conjugation and safely excreted. Even in a therapeutic dose, however, a small amount is metabolized by the P450 system to produce the toxic metabolite NAPQI. In a small amount, NAPQI is safely detoxified by cellular glutathione. Now, in overdose, we see that a large amount is formed by the P450 pathway, probably because conjugation is saturated. That consumes hepatocellular glutathione, and the NAPQI is free to bind to cellular proteins, particularly appears mitochondrial proteins, that leads to a cascade of oxidative stress and leads to hepatocellular death and, if severe, acute liver failure. Acetylcysteine, the antidote that we give, in fact, the only treatment we have, replenishes cellular glutathione. So clinically, what I see as a doctor is a spectrum of paracetamol injury, which very much depends on the time from the person taking the overdose to presenting and starting treatment. So on this slide, I've broken down a patient into somebody who presents soon after overdose, an early presenter, and that's within around 8 hours of taking the overdose to come in and starting treatment. Now, those early presenting patients have a high paracetamol level in their blood, but their ALT is normal, and the predominant process that's going on in these patients is metabolism of the paracetamol. And in these patients who've presented within around 8 hours of the overdose, acetylcysteine, or NAC, is very effective. The real clinical challenge comes with the later presenting patients. So these patients present after, say, 8 hours, say 8 to around 24 hours after the overdose. They still have paracetamol in their blood, but they're starting to develop liver injury, and their ALT may already be raised. Here, the predominant processes, as well as paracetamol metabolism, are becoming oxidative stress and mitochondrial injury. And in these group of patients, there's a need for an additional treatment as well as acetylcysteine. And there are drugs such as fomepizole or calmangafolapir being developed that may be of use. And then as we go further down the disease progression, we see liver injury is established, even liver failure. And in these patients, the paracetamol is metabolized. There's often none in the blood. And here we're looking at a complicated process of inflammation, cell death, cell regeneration. And it's likely that the treatments there are going to be anti-inflammatory or pro-regenerative therapies, but quite different to the early presenting patient. So in terms of biomarkers, I think the starting point always is, what is the unmet clinical need? And for this, I'll talk a little bit about the frequency of paracetamol overdose. So on this slide on the left, we've got the epidemiology in the UK. And on the right, we've got the number of paracetamol overdoses in various countries in Europe and the US. Now, if we look to the right, what we see is that the UK has got much higher number of paracetamol overdoses than the other countries in Europe. In fact, now that the UK has Brexited Europe, paracetamol overdose is an orphan disease in Europe, whereas before it was not, because it was so common in the UK. And again, although it's a problem in America, the UK actually has more cases than America. And given that our size is much smaller, it shows how much more frequent paracetamol overdose is here. So in the UK, it causes around 100,000 hospital admissions, and around half of these per year need treatment. We still have around 200 deaths directly due to paracetamol, and around 350 people registered for a liver transplant. So at least in the UK, if not worldwide, it is a major clinical problem and a common clinical problem. And the challenge we face in hospital is that patients typically arrive in hospital soon after the overdose. So a typical story would be a patient takes an overdose at a moment of crisis and then immediately tells somebody they've done it. It's an expression of how angry somebody is with somebody else. Around 80% of overdoses in the UK are deliberate. So most patients arrive in hospital soon after overdose at a time before ALT, the classic liver biomarker, will be raised. So what we use in clinical practice is paracetamol concentration in the blood to risk stratify who needs treatment and who doesn't. To go into that in a bit more detail, what we do is, and it's actually, when you think about it, quite crude, is we measure the paracetamol concentration and we ask the patient, how long ago did you take the overdose? And then we plot the paracetamol concentration against the time from overdose, and we have a line. On this slide, you can see the line we use on the UK on the left, the line used by most of the rest of the world on the right. So let's take some examples. Imagine the patient with the black dot. Let's call them Mr. Black. This patient has a paracetamol concentration of just over 100 at just over 4 hours, and they have a normal liver. Now, this patient is very different to Mr. Red, who's presented with a very high paracetamol concentration, late after overdose, and already has significant liver necrosis. However, what we do in the UK and globally is treat both these patients the same. They both receive the same dose of acetylcysteine, which is purely dependent on their body weight. And when you think about it, this can't be right. These two patients, one who's just got a level over the line, one who's got liver failure, are both only getting the same drug. And in fact, Mr. Black, if Mr. Black overdosed in the USA, he would actually be underneath the line and wouldn't receive any treatment at all, making the point that current management is not optimal. So what we need are new biomarkers. And I think there's an analogy here with myocardial infarction. So myocardial infarction, 20 years ago, a patient had a heart attack, they got chest pain, they were risk stratified by their ECG, and then doctors had to wait until cardiac enzymes were elevated. Interestingly, AST, being one of the cardiac enzymes, is also a liver marker. But now we have troponin, which is used routinely in clinical practice, which allows us to risk stratify patients at the front door. And I propose that's what we need for paracetamol. We have the history of overdose, we have the paracetamol concentration, but at the moment we have to wait until ALT is either elevated or not elevated to be able to discharge the patient or intensify their treatment. What we need are biomarkers that can move us much nearer to presentation. So the context of use, to use the terminology that I'm proposing, is a sensitive early biomarker of paracetamol overdose. What I'm saying is it should be detected within 8 hours of overdose, ideally. Patients at increased risk of liver injury despite receiving standard care. Now, there are a panel of biomarkers that are being developed by the consortium such as Safety and now by TransBioLime, and many of the audience will know them well. These include MIR122, keratin 18, HMGB1, GLDH, osteopontin, MCSF1. Now, they each have slight differences and read out slightly different processes. And for today's talk, I'm only going to talk about two of them, MIR122 and keratin 18, because these two are probably the lead candidates for my context of use. So MIR122 will be familiar to many of you, but it's a microRNA. And for those who may not know, microRNAs are short, non-coding, non-protein coding RNA molecules that regulate other mRNA transcripts and fine-tune the cell and are fundamental to cell processes. Now, MIR122, I put on the corner, is a freak. It's a freak because it's incredibly cell-specific. It really is only expressed in the hepatocyte, and it's expressed at extremely high concentration in the hepatocyte. So when the hepatocyte is injured, a huge amount of MIR122 is released into the circulation. So to summarize around 10 years of work, together with colleagues in Liverpool, we've shown that MIR122 is elevated in patients with paracetamol-induced acute liver injury, and that at first presentation to hospital, it can be useful as a biomarker. So I want to highlight this study that we published a few years ago now. So this was a study, quite a simple study, where we collected blood from patients when they first presented to hospital. So this is the blood that was taken after overdose to determine the paracetamol concentration. And then we followed the patient to determine whether they developed liver injury or not. And then we measured MIR122 and other biomarkers in this sample and asked the question, can these biomarkers identify who's going to develop liver injury early on before ALT is raised? And with MIR122, what we found was, so these are patients who have presented to hospital following paracetamol overdose with a normal ALT. So on the top left, you see 4 groups. There's a derivation and a validation cohort in the study design. And below the upper limit of normal, or 2 times above the upper limit of normal of ALT, is how we're defining liver injury. ALT was not different across groups, which is fact, because I'm only presenting here people with a normal ALT. Interestingly, paracetamol was also not different across groups. However, MIR122 at presentation was significantly elevated in those people who went on to get an ALT rise. And if we look at that with a ROC curve analysis, so a measurement of sensitivity and specificity, we see a very high performance. The ROC curve in both the groups was 0.96 and 0.97 in the 2 different cohorts. So highly accurate for identifying those people who are going to get liver injury despite current best treatment. The other marker I want to highlight is a marker called keratin 18. So keratin 18 is a protein that's expressed in multiple cells, but it's highly expressed in the hepatocyte. And the clever thing about keratin 18 is when it's released from a necrotic cell, it's released in a full length. However, when it's released from an apoptotic cell, it's released in a cleaved form. So you can use the ratio of the 2 in the blood to give some insights about whether a cell is dying by necrosis or apoptosis. The strength of keratin 18 is that paracetamol overdose causes massive cell necrosis. So we can use it to detect early cell death. And if we look at that same study I mentioned a moment ago, where we looked at the first blood sample, we see that keratin 18 is also elevated in those people who attend hospital after paracetamol overdose with a normal ALT. And keratin 18 has been replicated in multiple studies. So the table on the right shows multiple different papers. All of the ROC curves are above 0.9, the bottom one being alcoholic hepatitis. Point being that keratin 18 has been replicated, as has miR-122, but keratin 18 has been replicated in multiple different cohorts and shown to be a highly sensitive and specific indicator of paracetamol DILI. So at this stage, it's often useful just to give a real clinical case to sort of bring it to life. So this is all real. This is a 25-year-old man who took a large overdose of paracetamol at 2:30 in the morning. He'd written a message on Facebook, as is the modern way, and that confirmed the timing was 2:30, because if you think back, it's very important we understand the timing. He came to our hospital and was assessed 4.5 hours after the overdose. His paracetamol level was only 107. He had no risk factors for hepatotoxicity. His ALT was normal. So therefore, he was reviewed and discharged after psychiatry review. And that was entirely correct. The guidelines for the UK were followed correctly, and he was correctly discharged. He came back to hospital 43 hours later because he felt unwell. So his bloods are there in the table. When he first presented, his ALT was 34. When he came back, his ALT was 11,000. His INR, measure of liver synthetic function, had gone from 1 to 2.1. So this man was actually in a lot of trouble. He developed significant rapid liver injury. So we took the blood sample from his first presentation at 4.5 hours roughly after overdose, and we measured our markers, and we said, could we have picked up this man's liver injury and prevented him being discharged? And the answer is yes, we could, very clearly. His miR-122 was 50-fold above the upper limit of normal. His keratin 18 was 8-fold above the upper limit of normal. So this patient, it's a rare patient, but this patient would not have been sent home if these new markers had been used. And this gives an indication of how these markers may have clinical impact. Now, my remit was to talk about paracetamol DILI, but I thought it's important also to talk about non-paracetamol DILI. I just wanted to highlight two papers. This first paper was very much a landmark paper from consortiums in Europe and America that looked at non-paracetamol DILI and looked at biomarker performance. And all I want to highlight here is that keratin 18, again, comes out very favorably as a biomarker. miR-122 in this context is less favorable. So miR-122 may have more value with paracetamol DILI than non-paracetamol DILI. A study we recently published together with colleagues from Pfizer, we looked in this slide at the specificity of the markers. So here we're looking at different organ injuries. We're looking at healthy people, we're looking at liver injury, muscle injury, pancreas injury, GI and kidney injury. And if we focus on the second graph along, you've got keratin 18 showing high organ specificity. And if we look on the ROC curves at the bottom, we see that keratin 18 again comes out as the most organ specific. So within non-paracetamol DILI, it may be that keratin 18 is a lead above, say, miR-122 and others. So here's our biomarker pathway and the work we're doing. Thus far, we've used paracetamol and we have proof of concept and confidence that the biomarkers I've talked about have utility. So what we're doing now is trying to drive their clinical adoption in the UK. Something I won't talk about today, but is also to try to bring their use in resource-poor settings, particularly when people are being treated for tuberculosis, where DILI is a fear. We're also using them in drug development, and we're trying to use them in veterinary medicine, where there's a need because ALT is not a particularly useful biomarker. So one of the studies we're running now is we're developing a keratin 18 finger prick test, because what we need in clinical practice now is a rapid assay. Patient presents after a paracetamol overdose. We need to rapidly identify those people who have liver injury. And so together with colleagues at the University of Strathclyde and funded by the MRC, we're developing a finger prick assay, a lateral flow assay with a SIRS readout to be able to rapidly identify and quantify keratin 18 in finger prick blood to identify those people who have liver injury. And what we'll do here in this trial is say, okay, you're paracetamol overdose, you come into hospital, your finger's pricked. If you have evidence of liver injury, then you'll go down a different treatment pathway. One of the simple things we can do is start NAC immediately. Remember, NAC is optimally effective. It's started within 8 hours. But it also allows us to do novel clinical trials and identify a high-risk group to be given novel therapy, to do precision medicine. And briefly, one of the drugs we're developing, I'm not going to dwell on this, is a drug called calmangafodopa. It's a mouthful, particularly after I've had some dental work done this morning. But essentially, it's an antioxidative stress drug. And we've performed a phase 1 clinical trial with calmangafodopa, where we gave increasing doses of calmangafodopa to cohorts of patients. And we found that calmangafodopa was safe, but we also found, using keratin 18 that we've already talked about, there was evidence of drug efficacy. Even just in a very small trial, there was a significant difference in keratin 18 in patients who received calmangafodopa to those patients that didn't. Now, this is a secondary outcome. We did predefine it in the protocol, but it's a secondary outcome. But it's given us the confidence, using this novel biomarker, to go forward and do a pivotal multi-center trial. Now, before I finish, I'd just like to wrap up with a little bit of preclinical science we've been doing. And that is looking at where MIR-122 goes after it's released. Because we know that MIR-122 is released in huge amounts from the liver when it's injured. And we know that microRNAs can be taken up by other cells and can affect gene expression in other cells. So it's tempting to think that MIR-122, as well as being a biomarker, may also be a mediator. And what we found, and I won't go into too much detail, but I'm very happy to in the questions, is that in a mouse model where we deplete microRNA only in the liver using Crelox technology, we see that MIR-122 goes to the kidney. When it's depleted in the liver, it goes down in the kidney. When the liver's injured, it goes up in the kidney, but it doesn't go up in the kidney if the microRNA is depleted in the kidney. And we found that genes regulated by MIR-122 change in the kidney. So the hypothesis we're working up is that the release of MIR-122 from the liver has an impact on the kidney. And that may be a link between liver injury and kidney injury. So I'll go to my final slide and say where we are, but also what we need. So I think within the context of use I've described, early stratification of paracetamol injury, MIR-122 and keratin 18 have clinical utility. But what we need now are rapid, cost-effective point-of-care assays that we can take into emergency departments and actually test whether the measurement of these biomarkers in real time has an effect or could have an effect on patient outcomes. In non-paracetamol DILI, there's a panel of biomarkers, including keratin 18, being taken forward by the Trans-Bioline Consortium for regulatory qualification. And again there, I think keratin 18 has great promise, among others, to be used in non-paracetamol DILI. So what are the unmet needs? I think one of the biggest unmet needs that we have is to be able to identify or distinguish ALT rises due to DILI from ALT rises due to other causes, because none of the markers I've described are specific to DILI. They're specific to liver injury, but not DILI. So therefore, that's a great challenge. And the other challenge we have is to be able to distinguish a benign ALT rise from potential acute liver failure. And it may be that keratin 18 has some utility there. That's something for the future. But I'll stop there. Thank you very much.
Brilliant. Thank you, James. And so that was a really great talk. So just to finalize the the speaking session for today, I'd like to introduce Paul Hayashi. And so Paul acted as a co-principal investigator and co-chair of the Causality Committee for the US Drug-Induced Liver Injury Network, and now works for the FDA as a drug-induced liver injury team lead. So with that, I'd like to introduce and introduce Paul for his talk. Thank you.
Thank you. Just do a sound and visual check. Tom, are you seeing everything, and can you see me?
Yes.
Hello?
Perfect.
Okay, great. So thank you for inviting me for this talk on causality assessment in the clinical setting, and I'll allude to, of course, drug trials and looking at new development tools. First, I don't know. Hold on. Okay. Are you still with me? Can you see me?
Yes. You can see and hear you.
Okay, great. So disclosures and disclaimers, just up front, this is just me talking. This is not the FDA. This is not reflective of any way of their opinions or regulations or guidelines. So the outline for the talk is fairly simple. I'm going to talk about the ongoing challenges of DILI diagnosis, current methods to set up for the new methods. We can't really talk about the new methods without knowing something about the current ones. But before I get started, I just wanted to point out, this is obviously going to be a little more of a clinical talk, but I think it's important to point out that if you listen to the prior two talks carefully, much is dependent on that great science they do is dependent on getting good cases. And particularly for, I think it's Dr. Dominic Williams' talk, if you notice, if you listen carefully, part of the Bayesian model he was using is the validation was based on EUROTOX score of risk. And what's that based on? Well, it's based on clinical reports in the literature, and that's based on clinicians making a correct clinical diagnosis. So what we do here in this clinical realm has direct bearing on everything in terms of helping the scientists do what they need to do so that they can develop tools and biomarkers and et cetera. So it's all interrelated, and I think that's just an important point to make. It's probably evident to most of the audience, but I just did want to make that. So what are the ongoing challenges? I put these sort of 5 things together and to give you an overview, I suppose. So firstly, as mentioned before, the clinical presentation of DILI is highly variable. It ranges from causing liver tumors all the way to causing just late portal hypertension, cirrhosis, and everything in between. I did want to mention a couple slides about the challenge of herbal and dietary supplements because they have some special challenges that have bearing, both for themselves as well as in drug trials. Now, as the prior talks have talked about, trying to get objective tests and biomarkers is in development, and there's doing great work there, but there's no widely accepted objective diagnostic test to say this is actual DILI. And partly because of that, we don't have really great gold standard for cases or controls. And therefore, down to the bottom here, it's just basically the diagnosis is based on clinical acumen and exclusion of competing causes. So the prior two speakers mentioned this a little bit. I wanted to give you a visual about how variable DILI can be. This is a nice graphic that was published a few years ago in Journal of Hepatology. The cartoon in the middle is basically what's sort of a normal liver under the microscope. But as you can see, all of this array of pathology, and they have various listings of drugs that can do these things. And I just picked out 3. You don't have to be a hepatologist or a pathologist to see that these are extremely different. And they all have manifestations that from this pathology, they have clinical manifestations that are also quite different. And this is part of the challenge when you talk about DILI. It's just a wide range across hepatology and disease. And to make matters a little bit more complex, you'll see that some of these drugs appear in different categories. For example, oxaliplatin appears in these 2, and actually it could appear in this box as well up here. And I can show you that a little bit. So, for example, oxaliplatin causes various forms, actually, of liver injury, ranging from this acute cholestatic or hepatocellular injury. And in this case, this is a 46-year-old. He died of acute cholestatic liver failure within a matter of weeks of onset. And if you look at this one, this is where oxaliplatin causes nodular regenerative hyperplasia, which is a structural change within the liver. Not a lot of fibrosis, portal hypertension. And in this 5-year-old showed up with a varicose bleed years after taking oxaliplatin. This person had no idea that the oxaliplatin had he had any problems with this drug until 3 years later. So you can imagine this is quite a challenge to try and develop a causality tool or diagnostic tool to take in this wide range. And unfortunately, it hasn't stopped. We continue to develop, we continue to find new phenotypes for DILI. And this is one of the more recent ones that's come to the fore. This is a DILI-induced sclerosing cholangitis. And for the non-clinicians, this is an MRCP. Gallbladder, stomach, the liver's up here. This is the duodenum. This is the biliary tree draining the liver. It's supposed to look like what I say is like a tree on a winter's day. Very smooth, very tapered, and no issues with blockages. If you compare this is pembrolizumab that caused bile duct injury. And this is actually a classic picture of what we call idiopathic sclerosing cholangitis. As you can see, there's a lot of stricturing, there's upstream dilation because the bile can't get through, in a very diseased biliary system. The problem here is the latency is actually fairly long, and this is a cholestatic injury, and it's unclear to us what the natural course of this problem will be. So what about herbals? I think it's important to understand that the herbal industry is quite large. It's estimated about 150 billion US dollars worldwide. So you should not be surprised if your patients or subjects in a trial may want to or are taking herbals. The problem here is that there's been several now registry studies from across the globe, the United States, Spain, China, and Latin America, that shows that herbal-induced liver injury is not trivial. The rates run 20% in the US registries and the Spanish registry, all the way up to 27% in China. And the most recent publication from Latin America runs at about 16%. The reason these are split is often we split them into anabolic steroid type injuries and non-anabolic steroid herbal industries. So part of the reason I bring this up is in my new job at the FDA, I have come across this where I'm asked to look at cases within a trial, and there's actually no mention of herbals as a pertinent negative or pertinent positive. Or sometimes there is mention, but there's very little detail. I get lists of medications, but the herbals are kind of just hardly mentioned. And I think it is a problem because I'm left to decide, well, was there a background herbal in that case? And I have to decide and assess whether it's due to that drug or not. The study drug. So I think this is worth mentioning. And I think part of the problem with studying herbal medications, there are many, but I think this is probably one that is most troublesome to me. This was done by the DILI. When we have an herbal injury, we ask the patient if they would please give us the product so we can analyze it. And all these collected products are then sent to University of Mississippi, where they are analyzed by high performance liquid chromatography. And the point here is the mislabeling in these herbals and dietary supplements. So keep in mind, all these cases were actually a liver injury. The level of mislabeling is rather startling, and even the lowest at somewhere around 33% would be quite unacceptable, of course, in the drug development space. So I like to say this is, in terms of research of this problem, this is like we can't get past square one because we don't even know exactly what's in these products. So this is, again, part of the complexity on the clinical side of diagnosing drug-induced liver injury and its phenotypes. So what about current diagnostic methods? Well, of course, the sort of the most prominent, widely used is the RUCAM. Now, the RUCAM is a diagnostic scorecard based on clinical data. It was put forth in 1993, a long time ago, by a handful of very astute hepatologists and experts in drug-induced liver injury. It was not data-driven. It was basically their expert opinion of what a diagnostic tool should look like and the scoring system they came up with, what kind of clinical information should be included. And they came up with this scoring system. And it has done very well, but it is not perfect. So the strengths and limitations of the RUCAM have been written about quite a bit over the years. The strengths, it has objective scoring for timing, latency, and D-challenge. One of the biggest benefits is it covers the major areas of DILI diagnosis. So for a clinician, it's a very nice guideline. It's relatively simple, and it's the most widely accepted and published method, as I mentioned. But the limitations are important. There's a fair amount of subjective interpretation. So therefore, user-to-user reliability is not good, not terrific. Some of the variables are unclear whether they're needed. They don't have clear biologic plausibility. And it's not evidence-based, as I mentioned. And I just want to make this point to be clear. It's not designed, nor was it tested for drug development in the drug development space. So many of you know the RUCAM. I won't walk you through in detail of it, but I wanted to give you an overview. There are 7 criteria: latency and D-challenge. Basically, you fall into these time frames, 5 to 90 days, you pick up so many points. I wanted to just have you remember this point range of 1 to 2. I'll come back to that. And the D-challenge is similar. Excuse me. Sorry. D-challenge is similar, picking up points as you go along. I highlighted in yellow the ones that I think are a little problematic. This is risk factors, age, pregnancy. It's a little unclear that whether these really help with causality. Again, the biologic possibility was never clear to many of us. These are criteria 4 through 7. And here you pick up some other problems with reliability and subjectivity. So in the RUCAM, if you have the one drug and 2 other drugs that you're looking at, the fact that there are these other drugs will hurt the case for any one of them, so you can lose points. But the problem is the terms used in the RUCAM is, for example, known hepatotoxin. Well, what does that mean? That can mean different things to different clinicians or people. The other diagnoses, these are grouped into most common and a little less common, group 1 and group 2, and you're supposed to rule them out. But the problem is they don't tell you exactly what that means. It's up to the user to decide, did I convincingly rule these out or not? And again, there can be variability between users. And then DILI risk, this is one of the more problematic ones. It was based on labeling and publications. Well, again, there's a variation in how deeply did you look for the publications, what were the quality of the publications, et cetera, and how does the label, does it read, does it mean, is there a box warning? Is it just a mention of a mild elevation of liver enzymes? What does that mean and how do you score it? And then lastly, readministration of drug, this is a rechallenge. This is done fairly rarely. So I've always wondered how much it really helps with the tool because it's done, we challenge patients to give the drug again so rarely in practice and in trials, actually. So you add up all these score categories and they're bucketed into these 5 categories, ranging from excluded to highly probable. The performance numbers were published in the original article. I hesitate putting these in because frankly, I don't think that the controls and cases that they used were all that terrific. And even the original authors go through a lot of discussion about this. There was a fair amount of bias, for example, in the controls they used. But in any case, these are the numbers, performance numbers you have. So it's sensitivity, specificity for drug versus drug and for drug versus non-drug diagnoses. So not bad, but certainly not enough to be a standalone tool, either in clinical practice or in drug development. So moving on to the next most used or method that is out there is the US DILI, consensus expert opinion. So the DILI consists of about 25 to 30 hepatologists that have interest or expertise in the field of DILI. And I'll show you how we do it. Basically, we come up with, it's a subjective opinion in these 5 categories. And we always think in terms of percent likelihood. So we have discussions about, do you think it's at least 75% likely this was DILI? And we will come to a consensus to come up with these opinions. Where you may see this in drug development, well, these are the hepatology assessment committees or independent hacks that are that appear in drug trials, and generally they model it based on what the DILI does. So this is the cartoon of what consensus looks like, and I think it's important to understand the level of labor and time it takes to do this. So the patient is identified at an enrolling center. So the principal investigator will enroll the patient. And then there is a total of at least 6 months of time that rolls by of collecting data and follow-up data. So causality assessment in the DILI doesn't occur till 6 months after the event at least. All this data is collected. The data coordinating center then creates electronic files, a narrative, and a CRF. And then at a certain, Monday every month, these are released to 3 investigators per case. 1 is the principal investigator, and then 2 others independently chosen. They independently score the case. In this case, there was a disagreement between 2 and 3. And then on a certain Monday, 2 weeks later, every month, these are all released to the whole group, the scores and then a rationale. And then there's an email discussion for about 4 days. Now, if in that 4 days they can come to an agreement, then fine. It goes back and it's logged as a 2 here. If they cannot, then it is brought to a full committee call. There's can be lengthy discussions about the case. And if they still can't decide among the 3, it is brought to a full vote of the committee. And that's how this happens. So clearly this is not available to the clinician, but it is a fairly meticulous and robust means of coming to some consensus. So as I said, it's my opinion, and I think a lot of people's opinion, that doing validity on gold standard cases is difficult because there aren't gold standard cases that are unbiased. So we have not done a validity test, but we did do retest reliability, in which we took 100 cases, and these were old cases from a few years ago, and we basically assigned new reviewers, and we redid the whole process that I just showed you. And then we compared them to the prior scores. And when we put them in a 4 category score, because we felt definite and highly likely, or clinically and in drug development are going to be equivalent, we didn't do too badly. Actually, 73% of the time we came into the same category on repeat, 25% within 1 category, and only 2% were we off by 2 categories. So reliability is not too bad for this consensus expert opinion, at least within the DILI. Now, of course, you understand the difference between reliability and validity. What this study shows you is we are always hitting the same area. Now, whether we are actually on target, these are true DILI cases, that's, of course, debatable. We think we are. But again, we don't have great gold standard until we have a biomarker or something else that covers all these cases. Then this is what we can report for the DILI. So again, just to show you what I've done, I've kind of walked you through the ongoing challenges, sort of the current methods, and then we'll move on to talk a little bit about what are the new methods and where might this all be going. So one of the things that we've done, because the RuCam does pretty well, it's not bad, it just has some issues. A large group of us from the US DILI and from the Spanish registry embarked on this project to revise it and try to put it into a website application. This has been going on for a few years, and we're coming close to the end now. But the goals basically, I think up front, we wanted to increase the reliability so that when you got a case by a person, Dr. A, Dr. B would get the same score. We wanted instructions to be clear. We wanted to minimize subjective opinion. We wanted to make this as click and point and that's it as possible. We wanted an online calculator, and then we wanted to match the outcomes to at least match up against expert opinion in the two registries we had. So what did we come up with? Well, at this point, these are the major changes. This is the old RUCAM. As I said, there's 7 criteria. We changed things. Latencies, which I'll show you details on this, but the other big change was, if you remember, there were some factors that we weren't clear that they had biologic plausibility. Well, it turned out, yes, they fell out. They were early on in the modeling. They did not contribute to causality compared to expert opinion. So criteria 3 was eliminated. For now, we thought it was unfair and not valid to have a competing diagnosis scored within another drug. In other words, score them at the same time. We didn't think that did justice to each drug. So for right now, we have eliminated that. In other words, this tool is for one drug at a time. The other major change here, we realized that re-challenge happens so infrequently. We put it into a new domain of just additional data. It's not really necessary to have re-challenge to score this. So let me just show you some highlights. Latency scoring changes. This was probably the most data-driven changes we made because we tried to match it against the registry cases we had. If you remember, I told you to remember back in the RUCAM, the points range from 1 to 2. We stretched that range out to match the cases we had. But also, we also added this thing. This was Dr. Lucina was very astute in pointing out that we should have these stop points where if something is really quite completely inconsistent with DILI, then we should have the computer warn the user saying, hey, this is probably not DILI. You should not go on. Just stop and give your case a -6. Or if you go on, we're going to penalize you -6. And these are peppered throughout the tool. Onset from drug stop. Whereas in the RUCAM, this was scored and you could gain points the closer you were to the drug stop. We actually decided to penalize points. So you can only lose points. The longer you wait, the time period between drug stop and the onset of DILI, the worse it gets for the case for DILI. We kind of redid all the competing diagnoses to try to make them much more rigid so that you don't have this question, did you rule it out? We want you to enter from a drop-down list various test results, and then the computer will decide, hey, you ruled it out or you didn't. To give you an example, I give alcoholic hepatitis because I want to point out a couple things. The user just enters the AST and ALT at onset. The computer decides if this is even possible alcoholic hepatitis. If it is not, then this window won't even show up on your user interface. If, on the other hand, it does, and it fits some criteria to be alcoholic hepatitis, then the user is asked to pick from a very tight drop-down menu of these choices. And again, here's that -6 because the drinking was fairly heavy. We're saying, hey, this might be alcoholic hepatitis. Proceed with caution. We also tried to limit user subjectivity and entry errors so that on the website that's being developed, you don't have to calculate the latency. You just enter dates from a calendar. Domain 3, this was that risk likelihood that this was a DILI toxin. We tied it directly to the liver tox. So now you just pick the drug. The computer will go find the liver tox category and enter the points for you. You don't have to subjectively decide whether this is a high-risk drug. And again, here's that drop-down menu. You have to choose what the serologies were that you got, and the computer decides, did you safely rule them out or not? So how did we do? So these are the two, the DILI group and the Spanish group. I'll focus on the right because it looks better, and I'll explain the left. Again, these are the, I'm sorry, I have a touchy pad. These are the expert opinions along here: unlikely, possible, probable, highly probable. And they separated quite well based on the RECAM score. The DILI didn't separate quite well, especially over here with the unlikelies. And I think the reason is the DILI, we don't bring cases in unless we think they're DILI up front. In other words, if we ruled it out with a hepatitis E antibody or something else, we don't even enroll them. On the other hand, the Spanish group, they get referred cases in that they quickly can see that this is not DILI, but they retain those cases in their data. And that's why I like to say the Spanish group has really good, bad DILI cases. Whereas the DILI, these are cases that actually fooled us up front. And I think that's why the RECAM has a little more difficulty with these cases coming in. So that's where we are with the RECAM. We have a manuscript that's being finalized, and we have a website that's being finalized. So hopefully we'll have that out the door in the coming weeks to months. Well, what else? What else can help us with these clinical causality assessment tools? I'm going to just highlight a few studies that I think are interesting and important. This was a recent one, again, from the DILI. This was the value of liver biopsy. If you recall, in a lot of now the DILI IQ guidances and things, biopsies are sort of just set aside. It's not core or central because there was a great deal of data that biopsy helped us with diagnosing DILI. Here, what we did is we took cases where the first author, he actually painstakingly redacted with a black pen all information related to the liver biopsy and thereafter, and asked us again to go through the whole process to assess that case. And we assessed our likelihood categories. And these are these light blue bars. And then, monthly call again, we were given the biopsy results on a teleconference with our pathologist who went through the biopsy slides, and we were immediately asked to rescore. And what you can see is from these blue bars, which is with the biopsy information, the cases segregate to the left and right. In other words, biopsy did help. These cases in the middle became more clear to us as more unlikely or more definite or highly likely DILI. So we're going to need to decide how and if we can incorporate this into a causality assessment tool. But it is important data. This is another study which came out a few years ago. I have not seen a follow-up study. I am waiting because I think it is very interesting and exciting data. This is a group that has developed a means of taking monocytes from the patient who had the DILI, and they have a way of culturing these monocytes to take on hepatocyte features. And then they take these monocytes with hepatocyte features and they incubate it with the drug in question and other control drugs and other controls. And they just do a straightforward lysis assay. I believe it's LDH. So, example, this is the positive control using 100% tween. They get 100% lysis. And then this is the vehicle down here at 0% in these spider diagrams. And in this particular case, it was indomethacin was the drug that caused the DILI. So this is very interesting data. They took a cohort of cases, which they used their expert opinion to decide if it was DILI or not. And over here, this is the RUCAM score, the old RUCAM score. It did reasonably well, but their MetaHep score did even better, and the cutoff was pretty good. So interesting data, whether this can become used in clinical practice and in development. It remains unclear, but we await further data. What about genetics? There have been a lot of genetic studies, a lot of GWAS studies. And I think what it's coming down to is it will not be obviously one or two genetic markers that are going to tell us that this was probably DILI or high risk for DILI. But it may be a combination of markers. And this was a large study done by a consortium of groups, including many on the call, And it came down to, they looked at 3 markers, 2 HLA haplotypes and a gene variant in this PTPN22. And as you can see, if you had all 3, and this is for amoxicillin clap, if you had all 3, your odds ratio went up to 13.8. P-values over here. So I think this is, again, not obviously ready for clinical use, or certainly not screening. It depends a lot on the prevalence of these mutations or haplotypes in the population. But can we get there? Can we get to a point where there will be some genetic markers that can help us with particular drugs in their diagnosis of DILI? Maybe one last example of this, because I think it's a good example, say, is sort of bootstrapping our way into better cases. This was, again, done from the DIL, and this was HLA-B 3501 for, this is an herbal, green tea-induced liver injury. Green tea extract, for those you don't know, can cause significant liver injury, and it can be severe, acute liver failure, the whole shebang. But in any case, we looked at our cases that we had already bucketed into our categories, and we looked at the penetrance of these HLA markers, and there was this sort of nice dose effect. Now, the numbers get small out here, but on the other hand, comparing it to the general population, other HDS, and what we call possible or unlikely, there was a definite trend that these markers were important. So what I mean by this is, now we're left with, as the Dylan group, because we get serum on patients and blood samples, we can get HLA on these cases, and we have to decide now, are we just going to incorporate that into our opinion? And this way becomes a better gold standard for these particular cases. And I'll finish up. This is my outline of where I took you around the survey of this field, what my view of it is. And I'll leave you with this slide, and I've added some arrows here. Sorry. So again, clinical presentation is highly variable. The herbal and dietary supplements are a special challenge. And we're left with this no widely accepted objective tests and no gold standard for cases in control. But can we sort of, and I think we are, bootstrapping our way into these defining better cases and controls, so that then we can limit or at least I should say help the clinician make these diagnoses with better tests. It'll always come down to clinical acumen and exclusion of competing causes, but I hope we can make it at least more clear-cut for them. And with that, I'll end. Thank you very much.
Excellent. Thank you, Paul. And so with this section of the seminar, I'd just like to open this up into the roundtable discussion. And so first off, I'm going to try and see if this works, but to see if there's some questions that have gone on into the chat function. So whether it be possible for some of the people who've asked those questions to again ask those questions live and get the responses from the speakers. So could I start possibly with, if it's possible, would you be able to unmute yourself and ask the question that you had in the chat again, please? And if not, I'll ask that question for you.
I think that Gerd is on mute actually, but I don't think.
It's,
okay.
Yes. So I had a question for Dom to ask about how 3D systems, would they be able to allow assessment of immune-mediated DILI mechanisms? And do you see any advances or possibilities to be able to do this in the future?
Yeah, hi, Tom. Can you hear me okay?
Yes, that's perfect.
Yeah, I've sort of answered a little bit of it in the chat. I think we're certainly getting there with respect to the innate immune system. If you include Kupffer cells in co-cultures, you pull out differential sensitivity to trovafloxacin. These Kupffer cells tend to be quite functional. They release IL-6 on response to LPS, and so they enhance sensitivity to certain potential DILI compounds. I'm not really aware of a model, certainly an in vitro model, that can do the adaptive immune system. The MetaHep system that Paul talked about is probably one of the ones that is going some way towards it, because those it's able to identify drugs that potentially cause DILI through an immune-mediated system in an in vitro model, but you need the mononuclear cells from the patient who already have the sensitivity factor. And so, really this is a very patient-specific thing, and, you really require, in order to detect those innate characteristics, the adaptive characteristics, you probably require the patient cells in order to do that.
Brilliant, thanks, Sam. And another quick question for you, sir. Another quick question for you from Maribel was, again, for your model systems, do you anticipate the possibility of being able to incorporate patient-specific factors in these systems?
Yeah, again, it's what sort of patient-specific characteristics are required. Certainly, we can develop non-alcoholic fatty liver disease hepatocytes, which can be used in vitro, which show enhanced sensitivity to some DILI compounds. But really, what we're trying to do at the moment is really capture the phenotype in these models, because NAFLD cells are meant to have differential cytochrome P450 activities. And so we're trying to characterize these to make sure we get the correct phenotype. On top of that, we're also developing NASH models, so introducing an immune stimulant into those systems, but they're not really ready for any sort of risk assessment at the moment. It's still characterization.
Thanks, Tom. And a question for James is, would you be able to elaborate on any of the target genes of MIR122?
Yeah, sure, absolutely. So there are a large number of genes that are known to be regulated by MIR122. The data I showed are from a mouse model that we've developed. And maybe I'll go in a little bit more detail about that. So that is the enzyme that makes microRNAs is called DISA. And what we have is a floxed DISA mouse. And we deliver CRE to the liver selectively with an AAV8 vector. So for many people, that'll make sense. For those people that sounds like gobbledygook, essentially what it is selectively cutting out the enzyme that makes microRNAs selectively in the liver. And what this does is deplete all the microRNAs in hepatocytes. Because MIR122 is the predominant microRNA, it then allows us to track where that MIR122 has gone to when it's left the liver. And what we see is it goes to the kidney. It doesn't go to other organs. So it selectively goes to the kidney. Now, in the kidney, then, we can look for which genes are regulated by MIR122. Now, that's always a trickier thing to do than it sounds, because it really depends on whether those genes are expressed in the target organ. But the gene that we found, or that is of interest, of course, is it regulates cytochrome P450, 2E1. So MIR122 is well known to regulate cytochrome P450, 2E1, and that is expressed in the proximal tubules of the kidney. And we found that when MIR122 was depleted in the liver, it increased in the kidney at the mRNA, protein, and enzyme activity level. So that's evidence that MIR122 from the liver is traveling to the kidney and having an effect. Of course, it'll be affecting other genes. And that's some ongoing work that we're doing now with single-cell sequencing to try to understand which cells, particularly in the kidney, are regulated and what pathways are regulated by microRNA from the liver in that model.
Brilliant. Thank you, James. Then this is probably a question which crosses through to all of you, which is, so there's a question in the chat about cytokeratin 18s and whether they've been able to, whether there's assessments of them in preclinical species. And it really, I think that could be expanded to all the biomarkers on, for example, can we use these biomarkers in preclinical settings? in both animal and in vitro models in the clinical diagnostics, and also what do we need to do in a regulatory system to get confident use of these markers for validating the presence or absence of an hepatotoxic signal for new compounds and new modalities. So maybe if we start off with the preclinical setting, about the use of these new biomarkers in the preclinical setting, Dom.
Yeah, certainly. So, we can detect MIR122 in animals and in vitro cultures. It's a really great biomarker. And keratin 18, yes, we can detect that in, there's ELISAs available for keratin 18. The full-length keratin 18 indicates necrosis, and the caspase cleaved indicates a mechanism of apoptosis. But obviously, in preclinical settings, we're looking at very generally single drug effects, and so if you've got DILI, it's probably down to, in an animal or an in vitro culture, it's probably down to the drug you've treated it with. I think the difficulty tends to be, and I'll obviously let the clinicians comment on that, is when you get into more mixed settings, and certainly I could think of oncology being a crucial area whereby you probably getting something like keratin 18 released due to the efficacious nature of the oncology agents. Yet how would you then prize apart a potential DILI mechanism from that?
Brilliant. And for James, it's again the same, that question about, is there anything in the clinical setting and routine clinical practice for the biomarkers?
Yeah, I think what's important with using biomarkers in the clinical setting is to be very precise about the context of use. So within paracetamol toxicity, compared to the field that Paul talked about, it's quite straightforward. You've got the history of paracetamol overdose, you've got the blood level showing a high level, and you've got a dynamic ALT rise. So you can be pretty confident you're dealing with a case of paracetamol. So as my talk showed, in that context of use, these biomarkers are very helpful. I think expanding out into other forms of DILI, and I think Paul would be better to speak about that than me. I think the biomarkers have uses, but I think they have to be well defined, the context of use you're going to use them in. I can imagine, for instance, them being valuable in monitoring for DILI in a patient who is on a drug with a known DILI liability. But I think perhaps Paul is a better person to speak about that.
Yeah, no, it's a good question. I can't speak in detail, but I can just tell you the FDA is well aware of these biomarkers, and I'll leave it at that. So, of the different organizations or companies that are trying to research these things. One thing I wanted to point out, though, is it was a very important paper that by Rachel Church, and Dr. Deere showed that for the non-acetaminophen, if you looked at the area under the curves, still at the top were ALT and AST. So you have to keep in mind that outside of the realm of APAP, these markers, the ALT and AST are still, they are biomarkers. We just take them for granted, but they are there and they do a reasonable job. The one thing I would point out, and this has been brought up time again, is there are some ALT and AST rises that don't mean much. We all know that. So the question remains is, are these biomarkers, for example, there's some that are indicative more of necrosis, not just leakage of ALT and AST. And would it be able to not only diagnose DILI early, but also, so to speak, save that drug in the sense that the ALT-AST rises, but actually it doesn't mean that much because there's no necrosis. So these are all things that are out there. And as I said, the FDA is well aware of it. Will it get into the regulatory space and into clinical practice? I think so. I think that it'll be these niches for specific drugs, like what Dr. Deere does. He's got really great identification data. APAP is, really great drug level history, epidemiology. If you get that, then I think these markers for those particular indications may have a similar playing field and a similar success rate. That's what I would say.
Brilliant. Thanks, all of you. And a question for Paul. When do you foresee an available test for biochemical or genetic to allow us confident diagnosis of DILI and an opportunity to move away from these causality assessments?
This is from Andrade, I tell you. Probably knows better than I. So, he has much deeper experience. I don't know. I think that my understanding is that these markers, it greatly depends on when you're talking two things. When you're talking about screening in a population, I think there's a long way to go because these markers aren't that prevalent. A lot of them, the population prevalence won't make it cost effective. But yeah, I think it will be helpful in the case where you have the DILI case before you, and you want some added data to say, yeah, I really think this is DILI. And I think we're closer than we think. I can't give you a time frame, but I gave that last example with the green tea extract because, see, within the DILIN, we're even now struggling. Is that data good enough? Can we bring that into our causality assessment tool? Can we just say, hey, let's get that HLA, and then it'll help us decide if this is DILI or not? And that is clearly, we're bringing a genetic marker into our process. And we're talking about it even now because there are other ones, as you know. And there's precedence for this. We all know that, for example, abacavir, it is very clear. The FDA guidance says you're supposed to get that HLA marker for looking for hypersensitivity for abacavir. So I think we're closer post-diagnosis of a case of DILI. We're looking at it, trying to figure it out. Population screening to decide whether to give the drug or not, I think we're much longer way off there. Thanks.
Thanks a lot. And another question for you, Paul, is, in the meantime, what causality assessment method do you recommend for assessing DILI, particularly in clinical trials?
Yeah, that's a good one. So as I mentioned, I wanted to make clear is the RECAM nor the newer RUCAM is not for drug development, and partly is one of the 4 or 5, one of the 5 or one of the 7 domains is to look at prior history of liver injury. Well, we don't have that in drug development, so that is not to be used, in my opinion, reliably in the drug development space. Now, as a clinician in the clinical space, yeah, would I calculate a RUCAM on the back of a piece of paper when I was looking at a tough case? Yeah, I would. And I think a lot of us do. So I think the RUCAM, the RECAM that we are developing now, I think it will be an available and valuable tool. It will not be a standalone tool. There's just too much variability in DILI. It still takes your clinical opinion, and if you need it, and often you do, to get a hepatologist in there to look at the case. So that would be my answer. Having said that, if we could develop a tool based on in vitro data to give us that pre-assessment risk in the DILI space, in the development space, I think that would be nice. It would be interesting to look at that in a causality tool in the trial space. Thanks.
Thanks, Paul. Question for Dom. There's a lot of noise about reduced refinement and replacement of animals in toxicology and toxicology testing. And a lot of this hope and expectation is on the advancement of these more sophisticated in vitro tools. Where do you think we are in that kind of line, and what kind of scope of improvements do you think we'll need to be able to take drugs into man with limited preclinical animal data?
Yeah, well, it's a great question there, Tom. I think, I'd love to be able to say, there's no need for animal testing until regulatory. That would be fantastic. The first time we use an animal is actually in the regulatory setting. But I think certainly there's some way to go in the in vitro models. We tend to analyze quite considerably for safety, yet we don't actually have that much pharmacokinetics within those models, understanding of metabolism, exposure, where metabolites go. All these sort of things are really not known, but have huge predictive sort of capabilities. So in the discovery space, often we don't often know all the mechanisms of DILI, and so sometimes we get a little bit surprised by what we see, and we're not able to explain it. I've experienced one project where pre-regulatory animal studies were carried out, and it seemed to turn out that there was quite considerable pancreatic injury, which actually sensitized the liver to increased liver injury, and we were never really able to explain that. But in the individual models, yes, you could tell there was a certain level of pancreatic in an in vitro pancreatic system, but there was absolutely no damage within the in vitro liver systems. And so there's things that crop up which, with the current knowledge we have, we just can't explain. And if we went straight into a regulatory study with that particular molecule, we would be forced to either fail that regulatory study or use more animals attempting to explain the findings of that study. So at the moment, I think there's still a little bit of a way to go, but it's a great aspiration, aiming for, the first time to use animals is in that regulatory setting.
Great. Thanks, Dom. James, just a question on some of the data that you showed. You showed MIR122 to look to be really good in a paracetamol setting and less so in some of the other DILI settings. Do you understand any mechanisms for that or reasons why that might have happened?
Yeah, I think it comes down to the sort of homogeneity of paracetamol. So in paracetamol, you're looking at cellular necrosis, and we're looking at early samples. And in that setting, you're seeing a huge release of MIR122 in a patient group that we understand and are well defined. I think when you look at other causes of DILI, then because there's such heterogeneity, then I think MIR122 becomes less useful. There's also the phenomenon that microRNAs levels are variable across people, and they're more variable than proteins. And that's something that you see with MIR122, but you see with all microRNAs. We've just done a study showing MIR126 is a vascular marker. But again, we see a wide variability across people and even within people. And I don't think we understand why that is. Some of it may be analytical because the technique we're using, PCR, has got variability inherent to it, both in the extraction and in the PCR process. So I think some of the ability of MIR122 falls away because of its variability as well. But I think in paracetamol, it's so effective because you've got a huge necrotic process and you've got a huge release into the circulation very rapidly. And you've got a very sensitive detection method, PCR.
Brilliant. Thanks, James. A question for Paul from Maribel. She's asking that once we have a suspected DILI drug, would it be possible to incorporate into these causality assessments and scales specific information that are known risk factors for the drug, such as HLAs and genetic information that would help increase the confidence in the diagnosis?
Yes, I think so, and that's what I alluded to. Once the event has happened, if you have these HLA associations, then I think that is we are closer to putting that into practice than we realize, and so for example, I'm sure Maribel's alluding to the augmentin or MOX-CLAV cases. So I think that as development goes on, but we need, and she knows this, that we need a fair number of cases to do this kind of data. And again, it comes back to the quality of those cases. But to answer your question, yes, I think that is coming. Everyone knows the flucloxacillin story with their test and the HLA association and that kind of thing. So the short answer is yes. I wanted to come back to, I think somebody, Raul, had answered in drug trials. I forgot to directly answer how do we do it. Well, it's expert opinion in drug trials, and we often, the pharmaceutical company knows this, and they will put together an assessment committee, and not much use of the RUCAM in that space at this point. Thanks.
Thanks, Paul. And another question from Dom from says, with regards to predicting adaptive immune-mediated mechanisms of DILI, what extent does the integrated model have value in this through indirect interpretation of potential initiating events or necessity predictions?
Yeah, well, great question. I think that sort of crossover between the innate immune system and the adaptive immune system is crucial for kicking off a series of events. And, what is the nature of those sensitizing molecules that signal from the innate to the adaptive? In vitro, it's going to be extremely difficult to pull apart. There's the need for allogenic cells. You need the same B cell, T cell, hepatocyte, all from the same patient, which are not necessarily available. You also need cells from a sensitive patient, because not everybody gets the immune reaction. So, it's a huge ask identifying those individuals, and then ensuring that you have material from them to pull out those types of reactions. As I say, the Paul mentioned about the MetaHeps system, which is probably the nearest I've seen to get that bridging across, that there is some sensitizing factor that when you create a hepatocyte-like cell from a monocyte, they are still uniquely sensitive to the offending drug. So I think we need to take those cells and start looking at those types of factors that Sean mentions. It can't really be done in its own right, but we need cells from sensitive individuals. But identifying those sensitive individuals can generally only be done because they've already had a DILI reaction.
Great. And a question for all is, we've listened a lot to the, and particularly focused on some of the damage markers. Do you foresee, and do we use markers of cholestasis in your respective settings, and how do we use these? So starting with maybe James with a clinical.
Yeah, clinically, all we use at the moment is alkaline phosphatase, and I suppose gamma GT. And we don't use, say, bile acids at all clinically. In truth, I don't have expertise in that area. But at the moment, alkaline phosphatase, gamma GT would be the two ones that we'd measure for a cholestatic picture.
Paul, do you have any insights on this in your setting?
No, I think that's right. I think, most of the attention in DILI is on hepatocellular injury, which is problematic, I think, because these cholestatic injuries are important. As Dr. mentioned, ALCPHOS, other markers like supporting markers like GGT are done. Pediatric realm, they use GGT more. But I think it's an important point, and I wonder with Dr. Williams, with his great science, whether they're looking at this, because there's some pretty important cases or examples. Pexidartinib, for example, it's out now. It was approved, and it's under a REMS for a severe cholestatic injury. How do we study that in vitro and things like that? So I'd be interested to hear what Dr. Williams says too.
Yeah, thanks, Paul. No, funnily enough, we do have experience of that. Obviously, in vitro, we do have the ability to do a number of tests there. We if we don't know it's a cholestatic injury, what we'll probably do is some sort of omic profiling, look at the transporters of bile acids to see whether they are either directly inhibited or downregulated. One of the things that certainly seems to be coming to the fore is you can culture cells in vitro and you actually get the canaliculi forming. And there's information to suggest that if you image that canaliculi and you get shape changes within the canaliculi, they should be like sort of spaghetti around the cells, but they can change and become quite circular. These types of changes are indicative of potential cholestatic mechanisms, which we then, we don't have a single strong cholestatic signature, but we're able to put multiple pieces of data together, such as transporter downregulation, protein downregulation, or direct inhibition, along with effects on the canalicula, as I've just mentioned. And it sort of builds up. It's like piecing together a jigsaw. Once you have those indications of potential cholestasis, you can then go to the clinic and start looking for not only alk-phos, gamma GT, but bile acids within the patients. If you start seeing the shuttling of bile acids from the bile into the plasma, there may be, again, more weight of evidence to build up to say that there is a cholestatic signal going on.
Excellent. So I think we've just got to time for the session at 3 o'clock. So I just want to finish this and close the meeting and say thank you for all the speakers for all the effort in putting up together the slides and their insights. And thanks to Maribel and Simona for all their help in organising this. And so thanks a lot, and hope to see you all again soon.
And thank you, Tom, for helping to put this together too. Thank you.